Aging does not begin when wrinkles appear or when energy starts to fade. It begins silently, long before perception notices change, at a level far smaller than awareness can detect. Deep within the body, every cell operates under constant pressure to maintain order against forces that naturally push toward disorder. Life depends on this balance. Aging emerges when that balance gradually shifts. Every moment, trillions of biochemical reactions occur inside the human organism. Oxygen fuels mitochondria to produce energy in the form of ADP, allowing muscles to contract, neurons to fire, and organs to function in coordinated harmony. Yet this same process inevitably produces reactive oxygen species, unstable molecules capable of damaging proteins, lipids, and DNA. The body evolved sophisticated antioxidant defenses to neutralize these threats, but protection is never perfect. Microscopic injuries accumulate, and over decades their combined effect reshapes physiology. DNA damage represents one of the earliest drivers of aging. Each time a cell divides, billions of genetic letters must be copied precisely. Repair enzymes constantly scan for errors, correcting breaks and mismatches, preserving genomic stability. However, repair systems decline in efficiency over time. Environmental stressors, ultraviolet radiation, toxins, chronic inflammation, metabolic overload accelerate this process. Mutations slowly increase, altering cellular behavior and weakening coordination between tissues. Aging is therefore not a single disease, but a systems-level phenomenon involving interconnected biological hallmarks. Scientists describe these hallmarks as genomic instability, mitochondrial dysfunction, loss of proteostasis, cellular senescence, stem cell exhaustion, and disrupted intercellular communication. Each develops gradually, reinforcing the others in a self-amplifying cycle. Mitochondria play a central role in this transformation. These organelles, descendants of ancient symbiotic bacteria, generate the majority of cellular energy. With age, mitochondrial DNA accumulates mutations and efficiency declines. Energy production becomes less precise, increasing oxidative stress while reducing available fuel for repair processes. Cells enter an energetic deficit despite abundant nutrients, leading to fatigue and reduced resilience. At the same time, proteins within cells begin to misfold. Normally, molecular chaperones identify damaged proteins and guide them toward recycling systems such as autophagy and proteasomal degradation. These systems function as internal sanitation mechanisms, clearing debris before it disrupts cellular operations. When metabolic stress remains chronic, constant caloric intake, poor sleep, sedentary behavior, these cleanup processes slow. Waste accumulates, interfering with signaling pathways essential for survival. One of the most underestimated contributors to accelerated aging is chronic inflammation. Unlike acute inflammation, which protects against injury or infection, low-grade persistent inflammation quietly damages tissues over years. Immune cells release signaling molecules that, when continuously elevated, impair insulin signaling, damage blood vessels, and disrupt neural communication. This state, often called inflammaging, links aging to many modern diseases including cardiovascular decline, metabolic disorders, and neurodegeneration. Metabolism itself acts as a regulator of aging speed. When energy intake consistently exceeds expenditure, nutrient-sensing pathways remain locked in growth mode. Mechanisms such as mTOR signaling prioritize cellular expansion rather than repair. While growth is essential during youth, continuous activation later in life reduces longevity, pathways responsible for maintenance. Conversely, periods of metabolic challenge activate repair genes that enhance resilience. The body evolved expecting fluctuation rather than permanence. For most of human history, food availability varied. Physical exertion was unavoidable, and environmental conditions demanded adaptation. Modern environments remove many of these pressures, replacing them with constant comfort. Biology, however, remains calibrated for challenge. Without intermittent stress, protective mechanisms remain dormant. Cellular senescence illustrates this paradox. Cells exposed to excessive damage stop dividing to prevent malignant transformation. While protective initially, senescent cells release inflammatory factors that influence neighboring tissue. As they accumulate, tissue function declines. The immune system normally clears these cells, but efficiency decreases with age, allowing them to persist and amplify degeneration. Another critical factor lies in epigenetic regulation, the system controlling how genes are expressed without altering DNA sequence. Chemical tags attached to DNA act like switches determining which genes activate or silence. Over time, environmental exposures and metabolic stress disrupt this regulatory pattern. Scientists refer to this as epigenetic drift, a gradual loss of cellular identity that contributes to aging across organs. Circadian rhythms also influence biological aging more profoundly than once believed. Nearly every cell follows a 24-hour timing system governed by light exposure and behavioral cycles. When sleep patterns become irregular or artificial light extends deep into nighttime hours, hormonal coordination deteriorates. Repair processes meant to occur during sleep become incomplete, accelerating physiological wear. Even psychological stress alters aging biology. Chronic activation of stress pathways elevates cortisol levels, redistributes energy away from long-term maintenance, and weakens immune regulation. Short bursts of stress enhance adaptation. Persistent stress erodes resilience. Taken together, aging emerges not from a single failure, but from cumulative imbalance across multiple biological systems. The remarkable insight from modern research is that many of these mechanisms remain responsive to lifestyle signals. Aging pathways are dynamic, continuously interpreting environmental information. The body is not simply aging because time passes. It is aging according to the signals it receives. The speed at which aging unfolds depends largely on how effectively the body alternates between growth and repair. Modern lifestyles heavily favor growth signals, constant feeding, prolonged sitting, chronic stimulation, while minimizing the conditions required for restoration. This imbalance shifts cellular priorities away from maintenance, gradually accelerating biological decline. One of the most powerful regulators of repair is metabolic switching. During fasting or extended intervals between meals, insulin levels decline and stored energy becomes accessible. The body transitions from glucose dependence toward fat metabolism, producing ketone bodies that serve not only as fuel but also as signaling molecules. These molecules activate genetic pathways associated with stress resistance, mitochondrial renewal, and enhanced antioxidant defense. Autophagy intensifies during these periods. Often described as cellular recycling, autophagy dismantles damaged components and repurposes their materials for rebuilding. This process removes dysfunctional mitochondria, clears aggregated proteins, and improves metabolic efficiency. Reduced autophagy is strongly associated with aging-related diseases, suggesting that periods of metabolic rest are essential for longevity. Physical activity acts as another powerful biological signal. Exercise introduces controlled stress to muscles and cardiovascular systems, triggering adaptive responses that strengthen tissue integrity. Muscle contractions release myokines, chemical messengers that influence inflammation, metabolism, and brain function. Regular movement increases mitochondrial density, improves insulin sensitivity, and enhances vascular flexibility. Importantly, exercise functions less as calorie burning and more as genetic activation. It signals the body to invest in durability. Without this signal, muscle mass declines, metabolic rate slows, and resilience decreases. Temperature exposure provides additional adaptive stimulation. Brief cold exposure activates thermogenic pathways, increasing metabolic efficiency and promoting mitochondrial activity. Heat exposure induces heat shock proteins that stabilize cellular structures and assist in protein repair. These responses reflect ancient survival mechanisms still embedded within human physiology. Sleep remains one of the most critical yet underestimated longevity factors. During deep sleep, the brain's glymphatic system clears metabolic waste accumulated during wakefulness. Hormonal signals coordinate tissue repair, immune recalibration, and memory consolidation. Chronic sleep restriction disrupts glucose metabolism, elevates inflammatory markers, and accelerates cognitive aging. Even modest improvements in sleep consistency produce measurable benefits in metabolic and neurological health. Nutrition influences aging not merely through calorie quantity, but through nutrient signaling. Micronutrients function as cofactors in enzymatic reactions essential for DNA repair and antioxidant defense. Deficiencies impair cellular resilience long before clinical symptoms appear. Adequate intake supports biochemical processes that maintain structural integrity across tissues. Equally important is avoiding chronic metabolic overload. Excess refined carbohydrates and ultra-processed foods create repeated spikes in blood glucose and insulin, promoting oxidative stress and glycation, chemical reactions that stiffen proteins and impair function. Over time, these processes contribute to vascular aging and reduced tissue elasticity. Social connection and psychological meaning also influence biological aging. Human physiology responds strongly to perceived purpose and emotional stability. Positive social environments reduce stress hormone exposure and enhance immune regulation. Longevity research increasingly recognizes psychological well-being as a biological variable rather than merely an emotional one. At the molecular level, longevity pathways converge on energy sensing systems that monitor nutrient availability and stress exposure. Proteins involved in these pathways regulate DNA repair, inflammation, and mitochondrial performance. When activated appropriately, they enhance survival during adversity. When suppressed by chronic excess, degeneration accelerates. Aging, therefore, reflects cumulative signaling patterns rather than irreversible destiny. Cells continuously interpret behavior as instruction. Consistent signals promoting repair allow biological systems to maintain function longer than previously assumed possible. The most profound realization emerging from modern science is that aging speed is adjustable within meaningful limits. While genetics establishes boundaries, lifestyle determines how rapidly those boundaries are approached. Small daily actions compound across years, shaping cellular health in ways invisible in the moment, yet transformative over time. The body remains remarkably adaptable even later in life. Mitochondria regenerate, metabolic pathways recalibrate, inflammatory markers decrease, and cognitive performance improves when supportive conditions are restored. Repair mechanisms do not disappear. They simply require the correct signals to reactivate. Aging accelerates when the environment communicates constant abundance, stress without recovery, and inactivity without challenge. Aging slows when biology encounters rhythmic variation, effort followed by rest, nourishment balanced with metabolic pause, stimulation paired with restoration. The organism you inhabit is not fragile by design. It evolved to survive uncertainty, adapt to stress, and rebuild after damage. When daily life aligns more closely with those evolutionary expectations, biological systems regain efficiency. Time continues moving forward, yet biological age responds dynamically to behavior. The difference between rapid decline and sustained vitality often lies not in dramatic intervention, but in consistent alignment with fundamental physiological principles. Your body is always listening, and every signal you provide becomes part of the story written into your cells.